Okay, so now we look at tool used in recombinant DNA technology. So actually there are 5 tools in recombinant DNA technology. So kita tengok dulu yang pertama. The first one we call as target DNA. So this target DNA contain gene of interest. Okay, so the gene of interest is from any source. For the example, from human, plants and also animal. And then the gene of interest letter will be insert into vector. Okay, the example of vector is plasmid. So kalau kita tengok dekat sini, secara ringkasnya, target DNA ni maksudnya ni adalah DNA yang kita nak. Kenapa kita nak this DNA? Sebab dia ada gene of interest. Contohnya kita nak hasilkan genes yang mana dia boleh kodkan kepada production of insulin. So that DNA dan juga genes yang kodkan kepada insulin tu, yang kita panggil sebagai gene of interest. So gene of interest ni boleh datang daripada pelbagai source. Contohnya dari human, dari plants and also dari animal. Selepas kita dah dapatkan gene of interest, so that gene of interest will be insert into vector. So yang ni nanti saya akan bagi contoh. Okay, so now we look at restriction enzyme. Second tools of recombinant DNA technology is restriction enzyme. So, what is the function of restriction enzyme? It will recognize and cut or cleave specific base sequence of DNA called restriction site of both DNA strand. Okay, so means that the restriction enzyme, the function is to recognize, dia akan identify ataupun kenal pasti and cut. Selepas dia dah recognize specific base sequence, dia akan cut at the specific base sequence of the DNA. And then tempat di mana dia cut tu kita panggil sebagai restriction site. Okay, and then it will produce DNA fragment known as restriction fragment. Okay, so saya buat notes dekat sini. RE refer to restriction enzyme. It will hydrolyze the phosphodiester backbone of each strand. So, based on the types of restriction enzyme, di sini awak perlu tahu dua contoh. The first one, saya bagi di sini adalah EcoR1. So, EcoR1 is commonly used. So, macam mana kita nak tulis EcoR1? So, E must be in capital letter and then CO. So, awak mesti kena gariskan Eco. And then R1. 1 mesti kena Roman. So, for EcoR1, it will cut the specific base sequence in staggered way. And then it will produce TKN. Okay, so this one awak kena ingat lah. Okay, it will cut in staggered way and it will produce TKN. Okay, so each of the restriction enzyme, it will recognize the palindrome sequence. So, kalau untuk EcoR1, the palindrome sequence is GAATTC. So, what is the meaning of palindrome? Palindrome means the base sequence of one strand read the same as its complement strand. So, kalau kita tengok contoh, since EcoR1 recognize GAATTC, so palindrome sequence means kalau daripada 5' to 3', the sequence is GAATTC. So means that in another strand which run from 5' to 3' also will read the same, which is GAATTC. So means that for EcoR1, dia mesti kena recognize this palindromic sequence. So bila dia dah recognize this palindromic sequence, so it will cut at the specific site. So kalau untuk EcoR1, it will cut in between bases G and also A. Ni untuk strand yang pertama. Untuk strand yang kedua pun dia akan cut in between bases G and also A. So once the specific base sequence is already cut by EcoR1, so it will produce the fragment like this. So yang ni yang kita panggil sebagai restriction fragment. And then the cutting pattern for EcoR1 is staggered. Staggered ni macam bentuk zigzag. Macam bentuk Z. So dia akan jadilah GCTTAA, and then untuk satu lagi fragment it will become AATTCG. So yang ni yang kita panggil sebagai sticky end. Okay. So, what is the difference with another example of restriction enzyme? Satu lagi example adalah SMA1. So, sama juga macam tadi. S must be in capital letter, MA, and then awak mesti kena gariskan. And then 1 mesti kena huruf Roman. Okay, so SMA1 will cut DNA in straight way. Maksudnya dia akan potong straight. And then it will produce blunt end. So for SMA1, it will recognize the sequence CCCGGG. Same as the second strand pun sama, CCCGGG. So for SMA1, it will recognize this sequence and then it will cut in between bases C and also G in both strand. So bila dia cut di tengah-tengah sini, so it will produce this fragment. So kalau awak tengok dekat sini, dia cut in straight way. So this one yang kita panggil sebagai blunt end. Okay, so means that for restriction enzyme ni awak mesti kena tahu example and also dia punya type of cutting pattern. Okay, and then how restriction enzyme is used. Okay, so kita tengok kepada third tools. Third is DNA cloning vector. So what is the function of DNA cloning vector? It will carry gene of interest into host cell in genetic engineering. Maksudnya, gene of interest tadi yang sudah di-cut by resection enzyme, it will insert into the DNA cloning vector. Okay, so the example of DNA cloning vector is plasmid. So plasmid is isolate from the bacteria. Okay, so yang kalau contohnya dalam steps in gene cloning, selalunya kita akan gunakan plasmid lah. Okay, second types of DNA cloning vector is bacteriophage. So bacteriophage is virus, and then third cosmid. The last one is YAC. YAC means artificial chromosome. Okay, and then soalan dia selalu akan tanya characteristic of DNA cloning vector. So how DNA cloning vector based on its characteristic. So saya dah listkan dekat sini. Number one, able to accept foreign DNA in multiple cloning site or MCS. And then number two, able to replicate freely in host cell. And then number three, possess selectable genetic marker. So selectable genetic marker example is ANPR. So this genetic marker can be found in plasmid. So apa tu ANPR? It is the genes that codes for resistance to antibiotics. Okay, so next we look at the tools number 4, okay, which are host cell. So what is host cell? It is the organism that used to receive recombinant DNA. Okay, maksudnya bila dah berlaku insertion in between gene of interest into the vector, so it will produce recombinant DNA. So the example of host cell ni dia akan terima lah recombinant DNA. So example of host cell is E. coli. So apa yang awak kena tahu untuk host cell adalah what are the characteristic of host cell. So number one, it will able to receive recombinant DNA through transformation process. Maksudnya host cell ataupun in specific E. coli, dia mesti kena ada ability untuk receive recombinant DNA. So macam mana dia boleh receive recombinant DNA adalah melalui proses transformation. So this one nanti saya akan explain detail dalam steps in gene cloning. And then number two, able to maintain the structure of recombinant DNA from one generation to another. And then number four, able to amplify the gene product from DNA recombinant. Okay. And then the last tools in recombinant DNA technology are modifying enzyme. So what is modifying enzyme? It is the enzyme that modify DNA. So for the example is DNA ligase. So what is the function of DNA ligase? It will join the separate DNA strand together by catalyzing the reformation of phosphodiester bond between them. Okay, so maksudnya tadi gene of interest will be insert into plasmid or vector. So macam mana kita nak seal in between gene of interest and also plasmid? It is by DNA ligase. Okay. So once dah berlakunya insertion in between gene of interest and also plasmid, so it will form recombinant DNA.